Hello. Oh, hello. Oh, sorry. Now it should be working, I think. Yeah. Mm, yep. Okay. Now it should be working and I should be live right now. Okay. Now please message in the chat if I am live. Okay. I think I should be live right now. Mm-hmm. Am I? Let me just check it. Okay. Just wait. Just wait. Actually, I'm new with this microphone, so yeah, I just made a mistake. So मैं इंग्लिश में नहीं पढ़ाऊँगा वरना मैं जनरल इंग्लिश में पढ़ाता हूँ। लेट मी चेक कि मेरी आवाज़ कैसी है। मेरी आवाज़ आ रही है? स्ट्रीम दोबारा कर दी प्लीज़। आज ऐसा सब मत करो यार, आज ही तो अच्छा नहीं चल। Also tell me one more thing, जो स्ट्रीम है वो स्टेबल है या नहीं, ये और बताओ मेरे को, बिकॉज आई थिंक बहुत लैग कर रही है, मुझे पता नहीं है। सो यू हैव टू टेल दिस थिंग, क्या ये स्टेबल है या लैग कर रही है। आई विल वॉच द कमेंट्स। ओके, जस्ट टेल मी कुछ इश्यू है क्या, क्योंकि मुझे फील हो रहा है कोई इशू। ओके, देयर आर इश्यूज। आई विल सी। Okay, tell me this thing as well. Is the stream stable? I think it is. I mean, just let me play with the screen for once and then we'll start. Is this stream stable? Yeah, okay, it's stable I think. And अगर मेरी आवाज़ आ रही है तो मैं अभी स्टार्ट कर रहा हूँ, ठीक है। I am Aishwan, as you know. I am Aishwan. I am a medicine student in Luigi Benvitli, Campania, which is situated in Italy. I am from India. Okay, and today we are going to study about the cell. And I would say that there are no there's no aim today. We will be aiming for maybe a lecture of maybe an hour or of maybe more, and also expect these sometimes these typing mistakes because the screen it just overreacts a lot of times. Okay, just sorry. Let me fix one more thing as well, then we'll start the lecture. Yeah, now we'll start it. Okay, so we are going to start from the topic cell, and you know what? This paragraph, especially these points, partially permeable, freely permeable. I mean, they are a very fascinating. words. Now, if you ask me why they are fascinating, why do I think that they are so special? So the answer will be when you look at a membrane. I am not talking about a cell membrane. I am talking about a simple membrane. Okay. So in that case, there are mainly three types. First one being permeable, and in permeable, it will be a freely permeable, and the second one will be partially permeable. Okay. फ्रीली परमेबल इज द मेम्ब्रेन जो अपने अक्रॉस किसी भी चीज को पास होने देता है विदाउट एनी प्रॉब्लम. Okay, there's no problem in passing of one material from one direction to another. But yeah, it requires on the concentration gradient obviously. But in case of partial permeable, it do not lets all the particles pass through it. Why? There could be chemical reasons. There could be size, comparatively size reasons, like maybe big molecule cannot pass the membrane, but a smaller molecule can pass the membrane. Okay, but when we talk about our cell membrane, our plasma membrane, it is not partially permeable. It is not freely permeable. Okay, what is it then? It is sil- it is selectively permeable. Selective permeable. What do we mean by that? That our plasma membrane, it has ability to decide that which molecules will cross it as per requirement. Like when it requires, as we suppose glucose, it will do a simple machinery to take inside the glucose. And if it do not requires the glucose, then it will just block that machinery. Okay, and this nature, the nature of being semi-permeable, it is one of the most important nature of a organism to be alive. Okay, because it helps in controlling what a cell will have inside it. It can easily extract waste, and it can easily intake the needed substances. Okay, now we will talk about this paragraph that a cell can be thought of as a bag in which chemistry of life is allowed to occur. Yeah, like a plasma membrane, and inside the cell, I mean inside that plasma membrane, inside that bag, all the essential processes are occurring. Okay, partially separated from the partially separated from the environment outside the cell. The thin membrane which surrounds all cells is essentially in controlling exchange between cell and its environment. Yeah, plasma membrane, it controls which will go, what things will go inside and what things will go outside, right? It is a very effective barrier, but also allows a controlled traffic of materials across it in both directions. Right, like it will let some things go inside it and let some things go outside. Okay, sometimes it will do it with the same thing. Like sometimes it will intake the water because it requires the water. Sometimes it will release the water because it is as a extra material inside it. Okay. uh, okay, would you just tell me one thing? Should I teach in English or should I teach in like mixed language, English and Hindi? Just tell this to me, okay? I'll be teaching in that way. Please, please, just tell me this. Okay, though then I'll teach in English. Okay. The membrane is therefore described as partially permeable. As I just told you, partially permeable is, I would not use the partial permeable word for the plasma membrane. It is much more of a selectively permeable. And if it was a freely permeable, life could not exist. As I just told you, that the role of plasma membrane in sustaining the life is so huge. Okay, if it could not exist, if the plasma membrane could not have existed, if it was just a freely permeable membrane, life could not sustain, because the chemicals of the cell would simply mix with the surrounding chemicals by diffusion. Okay. Now, cell biology and microscopy. The wait, this topic is not necessary. I mean, yeah, biology is necessary, but you can just remember this. Nothing here like light microscope is not needed for you. So just remember that the study of cells have given rise to important branch of the biology, which is known as cell biology. Okay, and in cell biology, the study of structure and function of different type of animal cells and plant cells. It is known as cytology. Okay. Um, yeah. Now we will skip this topic and now we will come here. So here there are some diagrams of the like plant cells, but I will not teach this now. When we will cover them, when we will be studying about plant cells and animal cells, then we'll talk about this. Oh, what the fuck! Look at that, they are here. Okay, so then we'll have to take a look at that diagram. Okay. So, structure of a generalized plant cell, diameter about 40 micrometer, and it is seen by high quality light microscope. So, we'll not see too much of the detail, but we'll see some basic structures. Okay. So, here it is a plant cell, and we'll start from the outside. Okay. We'll start from the outside. When you look at the outside, here, what is this? This is the cell wall. Let me just, yeah. What is this? This is the cell wall. And when you go inside, a little bit inside, what do you find? You find this sausage-shaped organelles. You find this sausage-shaped organelles, which are mitochondria. Okay. Then you find these small circles like this, like this. They can be what? Lysosomes. Lysosomes. Okay. And then you look at this and these green rods. So when you think about the plant cell, what are the green things in the plant cell? What are the green things in the plant cell? Okay, let's take a guess. What are these? These are the chloroplasts. Okay, and in chloroplast there's chlorophyll is present. So these are the chloroplasts. Sorry, I'm sorry. Yeah, and as we all know that there's a very large central thing situated in a plant cell. What is it called? It is called a vacuole, a large central positions vacuole. Okay, which may require it may require as per requirement how much space? 90% space of the plant cell. Okay, and here we have the nucleus. In the nucleus, this is a thread-like material, which is what? Chromatin. And here's a densely stained material, which is what? It is a nucleolus. And both of these together, what do they form? They form a nucleus. Okay. This was the a very basic structure of the plant cell. And here, yeah, This is a Golgi apparatus. I forgot to tell you about that. Okay. And one more thing here, when you look at the cell wall of this cell here, you see another cell wall here as well. It is the cell wall of adjacent cell. Is it the cell wall of the cell we just studied? In middle of them, here, this black line, it is the middle lamella. Okay, and here these are called plasmodesmata. The function of plasmodesmata is to connect cytoplasm of two adjacent plant cells. Okay, and now we will talk about animal and plant cells, and first we will be starting from the common features in them. So, like, first of all, I mean, you know about these things, okay, because you have prepared for the need. So basically you know about these things, but we will still be covering them. Why? Because first of all, like, first and the main thing here, that when you look at the way things are mentioned here, because when you look at NCRT, it is like a mess, okay, and you are trying to find facts in the mess, okay. But when you look at here, like, you can see there are common points in one direction and there are differences in the another direction. So it totally helps us to differentiate things, and also if we can differentiate the things into one and another, then we can also remember them very much easily. Okay, I will differentiate the things, but also I will be making a list, so a sort of a list here. Okay, just let me common things. Okay. Okay, let's start now. Okay, so in animals and plant cells, both are surrounded by a very thin cell membrane. Yeah, of course, plasma membrane is present in every sort of cell. It is not just about plant cell or animal cell. Many of the cell connects contents are colorless and transparent, so they need to be stained to be seen. Okay, this thing I want to tell you, that when we think about a normal cell, or everything like tissue, muscle tissue, everything, so what we have in our mind, that okay, muscle cells have a red color, or this different cell have this color, but no, I mean, cells are basically colorless. Okay, the only way you can look at them is they are stained. Okay, you can look at a muscle cell because it is stained by blood, by that hemoglobin. Okay, but in reality the cells are not of any sort of color, they are like stained. Okay, when you look at a plant cell, and if it is green, it is stained because of the presence of chlorophyll. Do you understand that? And many of the cell contents are colorless and transparent, so they need to be stained to be seen. Each cell has a nucleus, of course, which is relatively large in structure, that stands intensely and is therefore very conspicuous. And, like, what do we mean by that? Okay, like, I think you also have studied for the NEET, and when I was studying for the NEET, I had also, I mean, I also always have this thing in my mind, that it is conspicuous. What do we mean by conspicuous, and what is the relation of being densely stained and conspicuous? Okay, so I will tell you that, um, When we do the staining, like these two things, okay, you are doing a staining, okay. Staining is basically filling colors, okay. So like you put colors in this thing as well, and you put same colors in this, okay. And then what will you do? You will wash them, right? Then after that you will find that this thing has much more colors, but this thing do not have any colors. They washed off with the water. What is the concept here? That inside the nucleus, inside this ball there are very tiny detailed structure, that when you put colors inside it, they were stuck into those gaps, they were stuck into those structures, okay. So even after washing, there were still colors, there were a lot of colors, okay. But in this case, there was nothing inside. So when you put colors on it and you wash them, they were clear, okay. It is the relation between being the conspicuous and also the stain, okay. If if a um, if your organ is intensely stained, it is densely stained, it means that inside it there are too much amount of details, too much amount of structures, okay, which makes it very conspicuous, or we say very detailed, or we say it has much more amount of structure inside it, okay. I hope you understood what I just said, but for me personally, it was a very peace of mind when I understood that thing, because otherwise I just used to revise like, okay, okay, yeah, yeah, I know that it is very densely and conspicuous, but I never knew the meaning of it. Okay, so it is intensely stained and it is very conspicuous. The deeply staining material in the nucleus is called the chromatin. So when you stain a nucleus and the material inside the nucleus which gets stained, what is it called? It is called chromatin. It is a mass of loosely coiled thread, mass of loosely coiled thread, and that thread is made of DNA. Okay. The material collects together to form visible separate chromosome during nuclear division. In the metaphase, during the nuclear division, it forms what? It forms chromosome by condensing together. Okay, it contains DNA, a molecule which contains instructions that control the activities of cell. Within the nucleus, and even more deeply staining area is visible, which is the nucleolus. We all know that the most dense region inside a nucleus is a nucleolus. Nucleolus do not have a membrane. It is just a very high densely stained region in the nucleus. Okay, which is made of loops of DNA from several chromosomes. Right? There is like a not an intersection, but it is like a mess where the DNA from different chromosome is like coiled around each other. Okay, the the number of nuclei is variable 1 to 5, being common in mammals. Um, the number of nuclei in cells, for like that, if I give you example, I hope you've heard about it, that in paramecium, it have two nuclei. Okay, so it is the example of a multinucleated cell. Okay. Also, I think camels RBC, if I am not wrong, I am not so sure about that. But paramecium, I am sure. Okay. The material between the nucleus and the cell surface membrane is known as cytoplasm. Cytoplasm is an aqueous watery fluid, uh, varying from a fluid to a jelly-like consistency. It will generally depend upon the hydration situation of the cell. Okay. Many small structures can be seen within it. These have been linked to small organs, and hence they are known as organelles. So In the cell, we just saw the structure that it is a cell. I'll just clear it. Sorry, I'll clear it, all of it. Yeah. Okay. When you look at the cell, there are small these organ types things. These are collectively called organelles. Okay, an organelle, it can be defined as a functionally and structurally distinct part of the cell. Organelles themselves are often surrounded by membranes so that their activity can be separated from the surrounding cytoplasm. So, I mean, there's a sorry, I was looking at something. Okay, let me just check the thing. Yeah. So, as I told you that a cell, its plasma membrane, it helps its content plasma membrane, it helps its content. Let me do it on a fair place. I mean, here it's not nice. Plasma membrane, what it does, it separate content from surrounding in cytoplasm. Just like that, in every organelle, it's done by that organelle's membrane. Okay, I hope you understood that. Because if the materials will be separated, the required material in that case will be at the required place, okay, instead of being around everywhere, okay. And doing this compartmentalization, it increases the efficiency by a lot. Now, what is this thing called? It is called compartmentalization, okay. Having separate compartments is essential for a structure as a complex as an animal or a plant to work efficiently, as I just told you. Since each type of organelle has its own function, the cell is said to show division of labor, a sharing of the work between different specialized organelles. I mean, it's a very basic thing that if every organelle has its own different area, has its own different content inside it, then they are doing their own work, and it is called division of labor. Okay, and it helps the plants and animals to work very, very efficiently. The most numerous organelles seen with the light microscope are usually mitochondria, and especially in a animal cell. Okay, mitochondria, they are just visible but films of living cells. Taken with the aid of a light microscope, have shown that they can change shape and divide. Okay, by the help of light microscope, we came to know that mitochondria is single mitochondria. It has ability to what change its shape. If it can change its shape, it can also change its dimension, its size, and it can also divide, and it can also move from one place to another. They are specialized to carry aerobic respiration, or we say formation of ATP by releasing CO2 and utilizing the how do I say, by utilizing the oxygen. Yeah. The use of special stain containing silver enabled the Golgi apparatus to detect for the first time. Okay, so it was done by Camille Golgi, and it was done by this silver containing stains, which is not so necessary, I'm telling you. The Golgi apparatus is part of a complex internal sorting, and distribution system within the cell. It is also sometimes called the Golgi body or Golgi complex. Okay, in Golgi apparatus, what happens that it is a sorting system, it is like a packing system or distributing system. What does it do? It gets substance from ER, okay, it will go to Golgi apparatus, it will either pack it, and then it will pack it and it will transfer it to required place as per requirement, okay. um, Okay, so here now I will tell you some things about IMAT. Okay, because like uh, like I have cracked IMAT, right? So there are, of course there are some things which I had in my mind, and I think they also gave me an upper edge, because of which I got selected, right? So first thing I will tell you that I think you also know it, because Parthi also have prepared for the IMAT, right? So IMAT is never IMAT is never a content heavy exam, right? In IMAT, there's not too much of content. What does IMAT focuses on? It focuses on logic. Okay, you have to be logical, right? You have to think. Okay, that what if this happens, then what will happen? Okay, you have to think. Like in IMAT, no one will ask you that what kind of membrane is a plasma membrane. Okay, they will not ask you about the permeability. What the fuck will they ask you? They will tell you that, they will ask you that what if it was a freely permeable membrane. Okay, but what could happen then? Was the life sufficient? Could life stand a chance then? No. But that's the thing IMAT will ask to you. Okay, IMAT will not ask you too much data. It will ask you how the things work. It will ask you that what if this happens, then what will its result? Okay, it will ask you, if this is happening, why the heck it is happening, right? So in IMAT, I'm always telling you, you need to be logical. Okay, always be logical. Never be don't miss any single thing. Okay. How do, okay, clear page here. नहीं करना था. Okay, this was the thing I wanted to tell you about IMAT. And yeah, now I would invite you to please ask me any question. I mean, it's been 20 minutes only, but still ask me any questions. If you have any sort of doubt, any sort of question regarding the lecture I have done right now, till now, I will, of course I will start it much further, but I'm just asking. If you have any doubts, just ask me. Until that, I'll just have a sip of water. Yeah, also, okay, tell me one thing. It is sort of a video. Also, it's not 20, it's 30 minutes. Okay. Tell me one thing which I wanna know, that like I am teaching you, but you have done it, right? So I want to ask that what should I keep the level? Should I be a bit less detailed and increase the speed or not? That's what I wanna ask. Can you hear me? It's good, but you need to increase it if, but you can increase it if you want. Okay. If the speed is good. I mean, like, what's the date today? It's 21st. Mm-hmm. And our, wait, okay, let's talk about the plan bit, right? Uh, Today is 21st of July, right? The exam is on 30th of September. Okay, you'll have to be a bit interactive now. I don't think we should. I think we should keep Sunday off, right? Obviously, like Sunday, it should be a cheat day. So Sunday should be off. So except that, if we consider, what will it be? Let me just check my calendar. Yeah. Mm-hmm. So, I mean, your exam is on 30th of September, and today is 21st of July. So, I am thinking that it's like a clear 2 months gap, okay? Because I would not. अरे भाई कर लेना भाई पब्लिक हॉलीडेज वो तो ऑफ कोर्स लाइक मैं उसी दिन पढ़ाऊंगा जिस दिन तू आएगा बिकॉज if you are here like I'll have a consistency right because अगर कोई नहीं होता तो there's no point of teaching because I know that here no one is listening but if you are here I'm explaining as much as I can okay so like we have 2 months of gap okay and the amount of chapters we have like main portion is the biology okay biology is the lengthy one okay except that physics it is a much smaller thing and if I remember like last time Ajinkya sir when was teaching us he completed physics in like a week or something so I don't think physics will be a headache and I know you are a bit scared of chemistry so we'll take care of the chemistry. Mm, so the main focus for me will be to cover the biology. I would love to cover the biology in 1 month. Okay. Also, I will be sometimes I will try to provide you some sort of mocks, which will be there will be basically some need PYQ and I need PYQs. That's what I'll provide. I can't make questions or something because I don't have that much time. I'd say. उसके अलावा maths and physics in one shot physics in one shot is possible but maths मेरा इतना strong नहीं है maths so I don't know that I don't think that I can teach maths but I can teach physics से भी because physics मैंने बहुत मेहनत किया हुआ है बहुत पहले से तो I can teach you physics but maths no maths you'll have to I would suggest you for maths you should go to ICSE ICSC 10th class I use this okay मैंने भी इतना efficiently यूज़ नहीं किया but आई एम सजेस्टिंग यू ICSC बोर्ड का 10th क्लास के जो आप यूट्यूब पे लेक्चर देख लो ठीक है आप क्या करो ना आप जाओ लोकोमोटिव मैं लिंक भेज दूंगा मैं ठीक है लोकोमोटिव की वेबसाइट पे क्या है ना सिलेबस मिल जाता है उस पे आपको जो भी मैथ में टॉपिक मिल रहे हैं आप वो टॉपिक प्लस जो PYQ के जो आपको टॉपिक है वो मेन उनको जो है तो फिजिक्स का वन शॉट ले लो भाई ऐसे एक दिन में खत्म कैन यू डू द फिजिक्स इन 1 डे आई मीन एट लास्ट तो फिजिक्स में मेन तो ये क्वेश्चन प्रैक्टिस ही आ जाएगा राइट वो तो तुम्हारे प्रेजेंस ऑफ माइंड पे डिपेंड करता है ओके मेन तो मैंने लाइक आई इफ आई रिमेम्बर इट तो लास्ट टाइम भी तुम्हें फिजिक्स में नहीं वो केमिस्ट्री में हाँ लास्ट टाइम भी तुम्हें केमिस्ट्री में दिक्कत था लास्ट ईयर इस बार भी तुम्हें आई थिंक केमिस्ट्री में तो केमिस्ट्री में आई थिंक आई वुड ट्राई टू गिव यू लाइक हाउ मच टाइम मे बी 2 weeks एट लीस्ट ऑल्दो इतना है नहीं केमिस्ट्री में कुछ छोटा सा पोर्शन है फिजिकल में टाइम लगेगा बस तो 2 से 3 weeks ट्राई करेंगे और अगर तुम बोल रहे हो कि फिजिक्स का तुम्हें एक सिंगल वन शॉट ही चाहिए तुम कह रहे हो ना एक दिन का ही चाहिए ना तुम्हें एक दिन में ही सारे फॉर्मूलाज वगैरह सब कह रहे हो राइट अगर ऐसा है कि तुम्हें complete physics 1 day like you want sort of a formula sheet or something I think so I mean it's possible बाकी नहीं लोगे तब भी नहीं चलेगा तब भी चलेगा ओ सॉरी या ओके तो यू आर द ओनली फकिंग स्टूडेंट आई गोट सो Yeah, I'll work according to that. So we have 8 weeks. So I'll try to complete bio in 5 weeks and chemistry in 2 weeks. Two नहीं, I mean 3 weeks. फिजिक्स का तो इट इज़ वन सिंगल डे ना, तो कोई दिक्कत है नहीं उसका, वो कभी भी हो जाएगा, ओके। एंड उसके अलावा लाइक उसके बाद तुम्हारे पास 1 वीक और बचेगा, 1 वीक में लाइक यू विल जस्ट हैव टू वर्क ऑन योरसेल्फ, ओके। सो हाँ ये तो सही है वैसे, 100%। ओके तो हो गया, ये डिस्कशन हो गया। ओके वी हैव ए प्लान। नाउ आई हैव टू जस्ट वर्क ऑन इट कि भाई मुझे कितना स्पीड करना है लेक्चर्स का, बिकॉज आई हैव टू वर्क लाइक हेल मे बी। Okay, now this part is done. Common things are done. Now we'll work on the difference. Okay, difference between animal cells and the plant cells. Okay, let's get serious now. Okay, um, the only structure commonly found in animal cells which is absent from the plant cell is the centriole, right? And plant cells also differ from animals in possessing cell walls, large central vacuole, and a chloroplast. Okay. So, you know that, I think, right? Because you are in it student. But still, I'll tell you that in animal cell, there's centriole present, which helps in the gamete transfer and the flagella formation and cilia formation, and all of these three things they are not present in plants, so plants will not have centrioles as well, okay. But in case of plants have cell wall, a large central vacuole, and chloroplasts, which all of them are present, sorry, absent in the animal cells, okay. And we'll talk about them now. मेरे ही भरोसे, हाँ कोई करनी है, I'll teach you। बस अपना और सब्जेक्ट्स देख लेना, लाइक 23 हो गया, प्लस 13 हो गया, एंड वो 7 हो गया। कितना क्वेश्चन हो गए? इट इज़ लाइक 10 3 1 4 2 3, राइट? इतने क्वेश्चन हो गए और कितने आते हैं? 60 क्वेश्चन आते हैं। तो अपने 17 क्वेश्चन का तो देख लेना, इनमें मैं हेल्प करूँगा, और ऑफ कोर्स सारा तो तुझ पे ही डिपेंड करता है। कोई तेरा कितना ही हेल्प कर ले, एट लास्ट तो भाई तेरे पे ही आने वाला है ना। कि मान ले पेपर में तू कैसे प्रेशर को हैंडल करेगा, तब तो क्या करेगा, है ना। एट लास्ट तो सब बात तेरे पे आएगा, ओके। या सो लेट्स वर्क नाउ, ओके, वर्क इज नेसेसरी। कैम में 15 आते हैं, ओके। 45 सेंट्रियोल। Under the light microscope, the centriole it appears as a small structure close to the nucleus, right? Even during the cell division, we see that during the early phases of the cell division, if it is the nucleus, centriole will be present here, right, on the opposite pole near the nucleus. Okay. Centriole are discussed on page 18. Will read it on page 18. Okay. Cell wall and plasmodesmata are very interesting topic. With a light microscope, individual plant cells they are easily seen than an animal cell because they are usually larger, unlike animal cells surrounded by a cell wall outside the cell surface membrane. So, by a light microscope, plant cells can be easily visible in much more detailed way as compared to a animal cell. Why? Because plant cell they have usually have large organelles. Large organelle. What do I mean by that? They have certain organelles like chloroplast, like they are large central vacuole. Okay, they are large in size, so you can easily take a look at them. But when you think about the animal cell the same, you found one different centriole, which is the fucking one different organelle, which is the centriole, which is a very small thing. Okay. They are surrounded by cell wall, yes. This is relatively rigid because it contains fibers of cellulose. Okay, the cell wall is rigid because it has fibers of cellulose. Okay, a polysaccharide which strengthens the walls. The cell wall, it gives a definite shape. What does cell wall do? It gives a shape. It keeps the cell from bursting or collapsing. Okay, it allows it to generate large pressure inside it, and it also helps it to prevent it. It also protects the plant cell from very large macromolecules. Okay, cell wall, it is a freely permeable membrane. It do not have any ability to control which things will go inside or outside like manually. Of course, according to the size of course of the cell membrane, it have cell wall it has, but not any other aspect, like not like the plasma membrane. Okay, plant cells, they are linked to the neighboring cells by the means of fine strands of cytoplasm, which are called plasmodesmata, which I just told you, which passes through the pore-like structure in their walls. Movement through the pores is thought to be controlled by the structure of the pores. Movement through the pores. Okay, so what do we mean by that? That if a pore is like a bit larger, then a large macromolecule can go through it. If it is a small pore, only small molecules like ions, like water molecule, like maybe a small lipid molecule can pass through it. Okay. And now we will talk about the vacuoles. So animal cells, they also have vacuoles, but they are very small. Also, they are temporary. Okay. So one thing like we also read in the need that animal cell, it is small plus multiple vectors. Okay, it is very small, very small. This small vectors. But in case of plant cell, it is one central very large vector. Okay, it is a permanent. These are just temporary. Okay, which are temporary structures. Mature plant cells, they often possesses a large permanent central vacuole. The plant vacuole is surrounded by a membrane, the tonoplast, which controls exchange between the vacuole and the cytoplasm. So the plant vacuole's membrane is a single membranous uh, membrane which is called tonoplast, and it has ability to control what will go inside the vacuole and outside into the cytoplasm. And what do we know about that? Know by that tonoplast, it also has a nature of partially permeable according to this book, which I consider as selectively permeable. Okay, the fluid in the vacuole is a solution of pigments, enzymes, sugars, and other organic compounds, including some waste products, mineral salts, oxygen, carbon dioxide. So in vacuoles, what are we storing generally? We are storing the substance first, like which are not required at that moment of time, and second, waste products. Okay. Vacuums help to regulate the osmotic pressure. Yeah, it helps to regulate the osmotic pressure of the cell, the flow of water inwards and outwards, as well as behaving in a wide range of other functions. For example, the pigment which colors the petals of certain flowers and part of some vegetables, such as red pigment of beetroots, may be located in the vacuole. Right? I don't think I have to explain anything in it. It is just pure fact. Now we'll study about the chloroplast. Okay, chloroplasts, they are found in the green parts of the plants. They are mainly in the leaves. They are relatively large organisms. As they are only seen with a light microscope, it is given possible to see tiny grains or grana. It is even possible to see tiny grains or grana. Okay, so the chloroplasts, they are green parts of the plant. They are relatively large organisms, as I just told you earlier. They have tiny structures which are called granum, multiple grana, inside the chloroplast using light microscope. These are the parts of chloroplast that contains chlorophyll. Chlorophyll is inside the granums, okay, or we say granum. The green pigments which absorbs light during the process of photosynthesis. Okay. Also, if any day like okay, now I'll remember. Okay, the main function of the chloroplast is discussed here. You can think, sorry, what the heck? You can you can think of a plant cell as being very similar to an animal cell, but with extra structures. Oh, in the first 19, I was like, where it is? Plant cells they are often larger than the animal cells, although cell size varies enormously. Right. Why? Because plant cells they have large size or values, so obviously they will be larger in the size. Do not confuse the cell wall with the plasma membrane. Yeah, both of them are different things. Both of them are different nature. Both of them have different functions. Okay. Vacuoles they are not confined to the plant cell. Animal cell they also have small vacuoles, as I told you, such as phagocytic vacuoles. Also, these are not usually permanent structures. What do you mean by phagocytic vacuoles? So phagocytic vacuoles are the vacuoles which take an outside material into the cell and destroys it and eats it. Okay, like it is done by I would say lysosomes. Okay. Uh, okay, this topic is not necessary. This topic is not necessary as I marked to say. Also, it is not necessary now. No, like all of it is just no. It is not necessary. No, nope, nope, no sir. Oh yeah, this diagram. I love this diagram. Okay. From here, yeah, from here the things will be necessary, but I will not be doing them today because I'm feeling tired. But this thing I will do it today because it excites me a lot. Okay, it is what is it? It is the ultrastructure of an animal cell. The fine detailed structure of revealed by an electron microscope is called the ultrastructure of an animal cell. Okay. Sir, he क्या और वो actually those things were related to microscope ना. We are not going to study microscope here. Okay, vector microscope and light microscope they are not necessary for the IMAT. Okay, we study as I told you. We IMAT is not data heavy. It is facts heavy. It is process everything. Okay, we'll focus on the all the important things. Okay, now we'll study only this diagram. We'll start from the inside. You look at the nucleus. It is the nucleus. There is a different densely strained material. It is called the nucleolus. I'm sorry. Now here, there are thin tubulic structure making a net. It is called the endoplasmic reticulum. Okay, and it is just a hover that the material is strained inside the nucleus. What is it called? It is called the chromatin. The layer of the nucleus, this whitish layer, it is called the nuclear envelope. Okay. Also the sausage shaped material, what are they called? They are called the mitochondria. Lysosome, the small green balls, what are these? These are the lysosomes. Golgi body, it is also present here. Golgi body, it is the Golgi body near the endoplasmic reticulum. What is it? It is the Golgi complex of the Golgi body. And ribosomes, I mean, we can't say ribosome, but we all know that ribosomes are very small in nanometers of size. Which book is this? It is the Cambridge A level. But you can use NCRT as well. Like you have practiced from the NCRT, you can use it. Okay, but if you learn anything new from the lecture, add it into that. Okay, if you learn anything new which you think that it might be important, you think that it might be a little bit more detailed, just add it. Okay, don't be a bit lazy. It can cost you much more things, much more important things. Okay? And here's the cell surface membrane. I don't think there's anything else in this diagram. That's it. So what do you want? Do you want me to continue it or what? Do you want me to continue the lecture? I mean, if you want me to continue, I can teach you a bit more. I mean, not too much, maybe Golgi up to Golgi complex. So, what do you want me to do? It is my longest lecture, by the way. Otherwise, I always stop at the 45 minutes. खाना खा लो क्या भाई या 15 मिनट. हाँ. I mean, okay, so we have two options that you and me, we can like pause the lecture for maybe 15 minutes or 30 minutes. Then, like for that time, we can eat something or we can take a break and then we can start it again. What do you think about that? And we'll start again. It will be maybe for like 30 minutes or 45 minutes a clean lecture again. No, I'm not busy at all. No. So tell me, 15 minutes more now, or take half an hour break and then we'll start the lecture again. Okay, 15 minutes more and call it a day. Okay, sure. Uh, okay, let's start. Okay. This was the diagram. I mean, it is a very detailed diagram, so I was doing it. And now we'll start the topic. Sorry. Hmm. Structure and function of the organelles. Compartmentalization and division of labor within the cell, they are even more obvious within electron microscope. Yeah, obviously, electron microscope gives us more detail and more information about the structure of the cell. Okay, the nucleus, it is the largest cell organelle. It is surrounded by two membranes known as nuclear envelope. Okay, as we all know that. Nuclear membrane, it is a double layer membrane. Okay, it is a double layer membrane. Also it have these pores which are called nuclear pores. Okay, now the outer membrane of the nuclear envelope is continuous with the endoplasmic reticulum. We all know that. Okay, the nuclear envelope, it has many small pores which are called the nuclear pores. They allow and control exchange between the nucleus and the cytoplasm. Example of substance leaving the nucleus through the pores are the mRNA and the ribosome for the protein synthesis. Example of substance entering the nuclear pore. Okay, notice this. Okay, substance leaving the nucleus are the mRNA and ribosomes, and substance entering the through the nuclear pores, these are the proteins to make ribosome, nucleotide, ATP, and some hormones such as thyroid hormone T3. Thyroid hormone T3. T3 is entering it. Okay, it is not for making that T3. T3 is entering inside the nucleus. Okay. Uh, Within the nucleus, the chromosomes are in a loosely coiled state known as chromatin, except during the nuclear division, उस stage में they are in a condensed form known as chromosome. Okay. Chromosome contain DNA, which except I mean, I don't know how to explain that because in a normal cell, the chromosomes are not visible. The DNA is just coiled as chromatin inside the nucleus, completely in the nucleus. Okay, but when cell division occurs, they get densely condensed and then they make a chromosome. Otherwise, chromosomes are not visible. Okay. Chromosome contain DNA, which is organized into functional units called genes. What does genes do? Genes controls the activity of a cell and inheritance. Thus, the nucleus controls cells' activities. It controls the future of the cell. And when a cell is about to divide, the nucleus divides first, so that each new cell will have its own nucleus. Also, within the nucleolus make ribosome using the information in its own DNA. Okay. Let me just drink some water. Okay. Endoplasmic reticulum and ribosomes. When cells were first seen with an electron microscope, biologists were amazed to see so much detailed structure. Like, yeah, when you look at the endoplasmic reticulum, you look at this different sort of details. Like, it is at a very microscopic stage, and there are so many details. Okay. The existence of much of this had not been suspected. This was particular to an extensive system of membrane. It is an extensive system of membrane running into the cytoplasm. Okay, like these are the membranes surrounding, making tubules, and they are running into the cytoplasm. Okay, and which was known as endoplasmic reticulum. The membranes form an extended system of flattened compartments. These are called sacs, spreading throughout the cells. Processes they can take place inside these sacs and separated from the cytoplasm. I'm sorry. Yeah. The sacs they can be interconnected to form a complete system, which is called reticulum. The connections have been compared to the way in which the different level of parking lot are connected by the names. They are like connected like when you go to a parking. So let me just do this. When you go to a parking, that when you look at that, one lane is here, and for the entry to the another lane, there's not like you cannot go just go from here to here, right? You have to go from path like this. Okay, then you go to the another lane, it has exit from there. When you then you go to the another lane, it has exit from there. Okay, these sort of connections are in the endoplasmic reticulum. Okay. The endoplasmic reticulum is continuous with the outer membrane of the nuclear envelope. Yeah. The endoplasmic reticulum, what does it does? That it starts with the surface of the outer nucleus membrane of a nucleus. Okay. There are two types of endoplasmic reticulum. First one being rough endoplasmic reticulum, and the second one being smooth. Okay. RER it is rough because its surface is covered with ribosomes. Okay, and they are visible as black dots in the figure 1.8. Like when you look here, when you look here, there are these. What the fuck is happening here? This layout has changed. I don't know how to change it, but okay. I mean. Mm, can I try something? I don't know how to do these things. I mean, okay, just leave it. Okay, so when you look at that, there are these black dots. They are representing the rough endoplasmic reticulums, ribosomes. Okay. Yeah, they are visible as black dots. At very high magnifications, they can be seen to consist of two subunits, one being the large and one being the smaller. And the ribosomes are the site of protein synthesis. In the cytoplasm, on the uh, endoplasmic reticulum, the ribosomes will be 80s, which will consist of 60s subunit and larger, and the smaller subunit will be the 40s one. Okay. They can be found free in the cytoplasm as well, and also on the rough endoplasmic reticulum. They are very small, only about 25 nanometer in diameter. They are made of RNA and proteins. Okay. Proteins made by the ribosome on the RER, they enter in the sex, move through them. The proteins are often modified in some way on their journey. Okay. What happens that from the ribosomes, proteins will be made, then they will be transferred inside the endoplasmic reticulum, and then they will transfer across the endoplasmic reticulum, and they will be kept modifying during that time. Okay. Small sacs called vesicles are a break off. The protein will be released from the endoplasmic reticulum in the form of what? In the form of vesicles. in the form of vesicles, and the vesicles will go and fuse with the Golgi bodies. Okay, and then they'll get more packaging. Okay, they will form the part of secretory pathway, because the proteins, they can be expelled from the cell by the Golgi vesicles. Okay, but in case of smooth endoplasmic reticulum, it do not have ribosome. So what will it do? It will do the formation of lipid and the steroids such as cholesterol and the reproductive hormones such as estrogen and testosterone. Okay, the Golgi body. Now we'll study about the Golgi body. The Golgi body, it is a stack of flattened sacs. More than one Golgi body may be present in a cell. The stack is constantly being formed at one end of the vesicle, which spread off from the endoplasmic reticulum. Sorry. The stack is constantly being formed at one end from vesicle, which bud off from the ear and broken down again at other end of the Golgi vesicle. So what happened that when material from the endoplasmic reticulum will be released, it will go on the Golgi body's one surface, and it will get forming, transforming into Golgi vesicle, go from one to another layer, another to another layer, and then it will be released as a Golgi vesicle. Okay. The stack of sacs together with the associated vesicle is referred to as the Golgi apparatus or the Golgi complexes. Okay, the stack of sacs there, like these stacks, these stacks, they together they are called Golgi complex, or we say Golgi apparatus. Okay, the Golgi body, it processes, and it sorts molecules. Okay, particularly proteins from the rough endoplasmic reticulum, because we all know that proteins requires much more of functioning in their processing. Okay, now for the ready for the transport in the form of Golgi vesicle, either to the parts of the cell or maybe outside of the cell, which will be called secretion. Two examples of protein processing in the Golgi body are the addition of sugar to protein, which will make them glycoproteins, or the removal of the first amino acid methionine from the newly formed protein to make a functioning proteins, or we say the activation of the protein. Okay. In plants, the enzymes in the Golgi body convert sugar into the cell wall compartment, which means it will make cellulose. The Golgi vesicle, they are also used to make lysosomes. Yeah, lysosome, the suicide hooking bags. Okay, now we'll only cover the lysosome. No, I think we can cover the mitochondria. No, function of mitochondria and we will not be able to do that. So we'll only cover the lysosomes. Okay. So, lysosomes. Lysosomes, they are spherical sacs surrounded by a single membrane. Okay, they have no internal structure. They are just filled with hydrolytic enzymes, which should be activated in acidic environment. I think. We'll see. Which must kept separate from the rest of the cell. Why? Because if it gets the required environment for its activation, it will cause the damage into the cell. Okay. Lysosomes, they are responsible for the breakdown of unwanted structures such as old organelle or even whole cell, or sometimes if there's a extra waste cell, it will be transferred into the extra waste substances inside the cell. It will be transferred into the lysosome, and lysosome will digest it. Okay, as in mammary glands after lactation or breastfeeding, and as in WBC, lysosome are used to digest bacteria, right? Enzymes are sometimes released outside the cell, for example, in the replacement of the cartilage with bone during the development. Sorry. For example, in the replacement of cartilage with a bone, like when a young baby is developing to an adult, suddenly its cartilage is being replaced by bones. Okay, so the cartilage will be destroyed by the lysosomes. Okay, and as in the head of sperm, like in the acrosome, to digest its part to womb by digesting its the coronary radiata zona pellucida. Okay, and I think this is it for our today's lecture, right? If you have any sort of doubt or any sort of queries, any sort of request, just type it. There's no issues. I'll totally accept that. कार्पे डीम भाई भाई इसका क्या मतलब होता है? व्हाट डज इट मीन्स? मुझे नहीं पता। नाइस क्लास। थैंक यू, थैंक यू। आई होप आई डू बेटर। कभी कभी मैं अटक जाता हूँ, सो आई ट्राई टू डू मच मच बेटर। ऑल्सो आई थिंक आई एम डूइंग गुड, बट आई विल ट्राई मच बेटर। ओके। और आपको कोई भी ज़रूरत हो, आई हेल्प यू। आई हैव गॉट नो इश्यूज। And thanks for joining the class. And if that's it, then I am ending the lecture. It will be the bye from me. Three people are watching. Huh? No. Size the day. Oh, seize the day. Okay, okay. Yeah, I see. I see. Okay, so that's it. Now we'll go offline. Bye bye. Take care and study well. Meet you tomorrow with the rest of the chapter. We'll try to cover it as soon as possible. Okay. Bye bye. Take care. Bye.